So welcome all of you to the May meeting of the Science and Engineering Council. I'm Mary Howe Grant, a past president, and we're very pleased to have you here. I'd like to mention that we have some new and renewing members. Thank you for joining. They are Tom Everhart, Bob Johnson, DC McGuire, and Jeffrey. I'm going to mess up this Polish name. We're very, very sorry. Sichkowski. I think we have some other board members with us today, and those are Yvonne de Graaf, Bob Lilly, Stephen Skopath, and John Fishpa. I'm really messing up names today. Sorry about that. I want to say thank you very much to those of you who donated to the Science and Engineering Council when registering for this event. The donations help with our ongoing admin expenses, and they also support our Science Fair awards. I also would like to say thank you to those who have been donating to Science and Engineering Council's scholarships that are given out through the Scholarship Foundation. We're glad you're with us, and I think you'll enjoy today's talk. Yvonne de Graaf, our program chair, will introduce our speaker.
Great, thank you very much, Mary, and happy to see all people here today. We have a full slate of upcoming talks scheduled through August, and hopefully we'll be able to return to in-person luncheon sometime soon. Our next meeting will be on June 8th. Dr. Rachel Street from the Las Cumbres Observatory will be speaking on hunting isolated black holes with microlensing and some exciting black hole discoveries that their organization has contributed to recently. On July 13th, Dr. Galen Moody from UCSB's Electrical and Computer Engineering Department will speak on quantum computing and quantum photonics. And on August 10th, Dr. Christopher Richardson from UCSB's Biology Department will be speaking on DNA damage repair. Today, we're very happy to have Dr. Kenneth Kozik as our speaker. He's the Harriman Professor of Neuroscience in UCSB's Department of Molecular, Cellular, and Developmental Biology. Professor Kozik is a neuroscientist who served as professor at the Harvard Medical School from 1996 to 2004, before moving to UCSB, where he is also the co-director of the Neuroscience Research Institute. He co-authored Outsmarting Alzheimer's Disease, co-founded the Learning and the Brain Conference for Educators, and conducted seminal research on Alzheimer's disease genetics and cell biology. His work in Colombia on familial Alzheimer's disease has appeared in the New York Times, BBC, CNN, PBS, and CBS 60 Minutes. His UCSB Arts and Humanities commencement address was archived at the Graduation Wisdom Best Commencement Speeches website. Professor Kozik is speaking to us today on the surprising science of brain organoids. You can post your questions in the Zoom chat or unmute yourself to ask them after Dr. Kozik's talk. Dr. Kozik, I'll stop sharing my screen and you can share yours.
Great, thank you very much. And now let me share screen and let me make this large. And here we go, let's see. Good, and I think everybody sees that? Yes?
Yes, but we don't see you.
You should. Let me see if I can.
Oh yeah, your camera's off.
Yeah, there we go. Now it's okay. Thank you. So I'm going to get back to the first slide here, previous. I'm going to talk about a development in cell biology that is the ability to make brain organoids that are based on stem cell biology and have taken advantage of the fact that stem cells, by definition, can differentiate into any type of cell in the body, including brain cells, neurons, astrocytes, all the diversity of cells in the brain. And that, you can, let's see if we can move this along. So just to give you a rough idea of that, we have over on the right, we have a brain here, which has which has these layers in it, and the layers are made up of different cell types, and those cell types, when the brain is developing, migrate out to the surface and further differentiate into mature neurons and glia. So when you start with a stem cell, now we're going over to the left side here, you can take these stem cells, and stem cells, as you may know, can be made from any of us now. There was a Nobel Prize given not too long ago to Yamanaka for the discovery that one can take a skin fibroblast or some cell from the body, put in certain transcription factors, that means proteins that control gene expression, and revert those skin cells back to a stem cell that has all the genes, that has all your genes in it. And once we now have those stem cells, we can grow them in a dish here where you see on the left, and they grow in a flat planar manner, differentiated into liver, kidney, all different types of cells, depending on the growth factors and how we guide them towards some destination, some desired cell type or tissue type. But the technology I want to talk about today is in the middle, cerebral organoid. And here, the discovery made by Madeline Lancaster not very long ago was to take the stem cells that were growing in a planar fashion in the dish and introduce them into a gel-like material that is something a little bit like Jello. It's called matrigel, and you take a drop of these stem cells, put them into the matrigel while it's a liquid, and then by changing the temperature just a little bit, it begins to form a gel. And when the stem cells, as they're differentiating to neurons, are doing so in a gel-like structure, they do something amazing. They actually begin to move into this third dimension, no longer flat and planar, and begin to assume some of the anatomical characteristics of the actual brain. They continue to make this diversity of cells, but they put all these cells in more or less the right place. They make this kind of laminar pattern that you can see here. Don't worry about these these letters. They're just the names of the layers, the lamina in the brain. And so you can see here that we have the lamina being reproduced in the organoid. These organoids are only maybe a half a centimeter. And to see all this organization, we have to slice them open, put them under a microscope, and there suddenly revealed is this amazing, these amazing patterns, which I'll show you here. So here's just a quick review of stem cells. I went very fast over this. Let me just go back one moment. You take these skin cells from any of us, put them in a dish, and put in all these words up here. These letters refer to specific transcription factors that are introduced into the skin cells, and they get reprogrammed. Reprogramming is the word that means reprogramming them, making them into stem cells. And the word we use is induced pluripotent stem cells, IPS. And as I said, they can be made into any type of cell. So back to the organoids, here's what they look like. They're just these little blobs floating in a dish. And they have this remarkable fidelity to brain organization, even though they're tiny. And there's a number of ways to make them. And I won't, a lot of this is quite technical. I'm going to jump down to the last point down here, where it says that they form even at the cellular level, not just the anatomical organization, but the actual cells themselves have synapses They have these structures called spines, which are little projections from the dendrites. They make inhibitory and excitatory cells. They really make a very, really complete repertoire. Well, not a complete, but a nearly complete repertoire of what we see in the brain. And if we now stain those organoids, and again, this is all background still. This is taken from the literature. Here is a brain that has been cut, and you can see this laminar organization with some cells stained red here, and then you can see different layers here in the green. These are, and down here, the black is the ventricle, the large area in the center of the brain that's just filled with liquid or cerebrospinal fluid. It's a technical term. And these and as the cells, as we move tangentially from the ventricular region out here, you see all this different organization, which is reproduced over here on the left in the organoid. Again, the cavity in the center and all this very, nice, let me just come into, I'm just, somehow things are now just going crazy. There we go. Okay, so, and by the way, I just want to emphasize what was mentioned at the beginning. Please interrupt me if anybody, I can't actually see you guys, so just unmute and speak up. So there we go. We can recapitulate cerebral organoids, recapitulate what we see in brain development. They remain rather immature. We haven't yet figured out how to get a completely mature brain. This says they mature up to about 24 weeks after conception. It's really gone beyond that now. We can make a brain, but it's still relatively, something like a brain, but it's still relatively immature. Now let's go down to actually looking at the cells themselves inside those organoids. And so here up again on the upper left is an organoid now, but this time it's stained the green color.
You all right?
Hello, the green color is, somebody should mute. The green color up there is marking the astrocytes. As you know, astrocytes in the brain there's probably more of them than there are neurons. They're supporting cells. They are supporting cells for the neurons, providing some nutrients, but they're also involved in a lot of the physiology. Astrocytes are very important cells. And you can see in the green in the upper left, there's lots of them. And as you look at higher power in that white box, you can see that they're quite complex. They look like real astrocytes with all of these processes around them. Let's go over to this middle panel called B, where now the green is something else. It's no longer an astrocyte. The green this time is the axon from a neuron, the long projection. And you can see that here in these organoids, there are long projections, long axons that are growing across the organoid. The blue in the center is relatively acellular. There's very few neurons there. That does not necessarily mean that it's forming that ventricular cavity I mentioned at the beginning. You have to remember the reason our organoids remain so small is because we have not yet figured out how to put a blood supply into an organoid. So as the organoid gets larger, the center part is deficient in nutrients. And so sometimes the neurons in the center just don't make it. Down here again, long axons here in the lower panel under B. And then panel C is very interesting because what we're labeling over here in C is a very important part of brain physiology. These are inhibitory neurons. You know, there's a large part of what the brain has to do is inhibition. And there are neurons that are dedicated to inhibiting the firing of other neurons. So those are labeled here. One is with a label called GAD65. Down here, another one of those inhibitory labels called parvalbumin. And finally, if we go over to these black and white panels or black and green and white panels, you can actually see the synapses in the organoid. Remember, synapses are how two neurons talk to each other. They exchange a chemical signal that goes across the synaptic cleft so that the electrical signal coming from one neuron can now be received by the next neuron after being changed into a chemical release from the presynaptic side to the postsynaptic side, and then reinitiating the electrical transmission. And that's basically, the synapse is the core part of the brain. It's really, the sort of the sine qua non of the brain. It really is something that is, each one of those synapses is the kind of microprocessor of the something like of the billions of neurons in the brain. People put the number at 86 billion. Every single one of the neurons has something like 5,000 to 10,000 synapses. You can just see how complex the wiring of the brain is. So what we're seeing here are these green dots or synapses along the dendrite of a neuron. And that suggests these are, that the organoid has reproduced a lot of the cell biology. Okay, there we go. So now let me introduce you to another technique here, which is getting very, very popular among scientists. It's a technology, it's a fascinating technology. This is called single cell sequencing. So you're aware now that we can sequence the genome. We can find out all the genes that are in the DNA. And as you know, from the central dogma of biology, it's a DNA to RNA to protein. So when the DNA is made into RNA, we call that transcription. And that transcription is and then when you go from the RNA to the protein, that's called translation. So we now have the ability to look not only at the DNA, but also to look at the RNA. That is, which genes are not just which genes are sitting, which genes are actually being converted into RNA. And the reason that's important is because, as you know, we know from the human genome that a human being has about 22,000 genes. But remember, the way cells differ from each other. Why is a brain cell different from a kidney cell? Well, the brain cell turns on some subset of those 22,000 genes, and the kidney cell turns on an overlapping but different subset of those 22,000 genes. So cell identity is conferred by which subset of genes in the DNA is getting turned on. That's how we achieve cell identity. And what we can do now, this is really quite recent technology, is to take any tissue, in our case an organoid, and separate out the single cells, individual cells, and see what RNA is being made in those individual cells. And that's what we're looking at here. Every one of these red dots is a single cell from an organoid, and it is then clustered in an unsupervised manner in which the clusters are separating these populations of cells based on the differences in the RNA transcription, in what RNAs are being made. So the computer doesn't know who these guys are. It just separates them because the brown is different than the green. Then we go back and we say, well, why is the brown different than the green? We see what genes are being made here. And we see that the genes that are in the green, for instance, are mostly astrocytes, because they're making genes now in the astrocytes. So we can colorize all the cells within the organoid. And you can do this for heart, kidney, brain tissue, and look at the populations of single cells that are present here. You can see all these green dots are astrocytes. Here's GABAergic here. The orange ones are inhibitory neurons. All the different cells, many different cells are present here. Okay, let's push onward. Please interrupt me if you have questions. A lot of this gets pretty technical. So And I'm just going to skip this because it's just showing more detail about the different cell types. Let's go now back to the organoids. Well, this is work. A lot of it when we decided to ask another kind of question. I showed you all the different cells in the organoids. We sliced them open, we stained them, we looked at the single cell transcriptional profiles. And now you can ask the question, we ask the question, what about their firing as neurons? What about their electrical activity? What kind of patterns are they finding? Is there some sort of encoding here of a neural code, a temporal code that people are very interested in investigating, how the brain takes input and converts it into firing patterns? That's a really fundamental question in neuroscience. So what we have done here is, here's the organoid over on the far left. And now what we do is we can slice it in this device. This is work that's done by a very talented postdoc in my lab, Tal Scharf. And let me just go back. Whoops, now I'm in this big problem again here. There we go. So we, what he did was to slice this organoid so that now it is no longer three-dimensional. It's a slice of about 500 microns embedded in agarose to help slice it. And here's the cool part: plop it down on an array, an array of electrodes that is made by a company called Maxwell Biosystems, which they've done some remarkable engineering. And what they've done here is to put into this surface, which has an area of just 2 by 4 millimeters, 26,400 electrodes with a pitch of 17.5 micrometers. So remember, a neuron itself is about 15 or 20 micrometers at most. So really, the electrodes here are pretty much corresponding to the size of the individual neurons. And so we lay a slice of this organoid down here, and those electrodes can record the activity from the neurons. And I'll show you here the next slide. Here's what it looks like. Go over to the far right, and we can record here we can pick up signals of different frequencies. They're using a high pass filter. We can actually now record individual spikes. That is when a neuron fires an action potential. Remember, after the synapse sends its signal to the neighboring neuron, it induces what's called an action potential. It's an ionic electrical propagation in which sodium ions flow in and create this electrical depolarization. And that is shown by the deflection here in the signal coming from an extracellular location, the extracellular positioning of the electrode. And then by doing a low pass filter, we can pick up frequencies in the brain that are much lower. This one, if you've ever had or been familiar with someone who gets an EEG, an electroencephalogram, and when we look at these human EEGs, you can't pick up this stuff, these action potentials. By putting electrodes on the skull, you don't see that. But we do see these lower frequency waveforms, and theta is a very common one. These waveforms change when you sleep and when you pay attention, and when you sort of, all different, there's a lot known about these waveforms. And we can see both the spikes here in the upper panel, and we see what are called local field potentials in the lower panel. Seeing all this in the organoids, we thought was really remarkable, that all of that is being reproduced. Let's see, I see a chat question. Why in the first instance did the stem cells made from skin, when put into the matrix cell, decide to make brain? Actually, so what they are is that while they come from skin originally, they're reverted back to a stem cell state before we do anything. And then once they're in the stem cell state, we guide them to a destiny of being in the brain or the heart or the liver. So, so maybe what I'll do is hold off on the chat questions till the end so I don't interrupt the train of thought. But if you have a question on exactly what I'm talking about, then please, again, just interrupt. And
Excuse me,
also-
what is,
yes,
what is electrode pitch?
pitch is the distance between the two electrodes.
Ah.
Yeah.
Thank you.
Good. So, and by the way, also, I wonder if the organizers here could also sort of help me when I get close to the end of this talk, because I can blather on for a long time, and I'll need to know when to begin to close down.
Sure, we'll do
Okay.
that.
Thank you. So here we go. Let's keep moving along. And now here we are looking at the firing in a different way. Here are, if we look at this is called a raster plot. And what we're looking at here is on the y-axis is time. So this organoid is cruising along, firing, and zero on the y-axis. Did I say the y? The x-axis is time. The y-axis is the individual electrodes. And this is a little bit of a technical thing because these are what are called local field potentials. So a single electrode could be picking up a signal from more than one neuron. So we have to also do something, I don't want to get into too much detail here, but we do a thing called spike sorting. So we can collapse all of the what we call units, the firing that comes from a single neuron, even though that single neuron is being represented by more than one electrode. So I told you there were 26,400 electrodes. We can route the circuitry in there and look at about 1,000 at a time. And then when we do what's called spike sorting, in this particular organoid, we have 131 different neurons that we're tracking. There are many more there. These are the ones we can see right at the surface. So every time you see a blue dot, that particular neuron is firing along that time period. And sometimes the red there, I'll show you these bursts. This is, again, very typical of the way brains and neurons behave, that there are population firing patterns in which many neurons fire at more or less the same time. And the details of those firing patterns are extremely interesting. So now let's look at the same thing in another way. So let's go over here, look at the blue on the left, and think that the blue is showing you the surface of that, what's called multi-electrode array. It's got all the electrodes down. This one may, I sometimes think this is one maybe I shouldn't actually show, but just to show you all of our data, this is one that actually has a rather large center in which there were no neurons. So that's why the center is all blue, but the color on the periphery, those sort of parentheses there, is where the neurons are. And you can see them now firing. The color is their firing rates, their spike activity. So some are red and firing very fast, some are slower. And this picture is really the XY axis here is the space. It's where each one of those neurons is in that two-dimensional multi-electrode array. And then if you go over to the right side, we can actually look at these spikes in terms of their amplitudes. Some have very high amplitudes and some are lower. Okay, now let's prove that these firings are really coming from neurons. So you do a little bit of pharmacology and you block with different drugs the firing of certain synapses. I'm showing that down here. I won't get into the particular details in the drugs, but we can actually prove that this is good neuronal proper firing because the appropriate drugs block the firing in the appropriate way. Now, here's something I think is pretty interesting. I hope it interests you as well. It's going to get a little technical, but it's I want to continue to make the argument here that we are we are really looking at features of firing that do replicate features of what happens in the actual brain. So here we're looking, if you look up in the upper left-hand corner, you see those letters called ISI. That stands for interspike interval. And the time between spikes, the distributions of the time between spikes is a parameter that, as neuroscientists, we're very interested in. Because the interspike interval, the distribution of those intervals, seems to differ depending on the anatomical location in the brain. Some places, the interspike intervals will be more stochastic and follow a curve that looks more Poisson, whereas other times they are more regular, more periodic. And if you look at these patterns here of interspike intervals, the interval is here on the x-axis and the firing is on the y-axis, you can see that we have put the mean over the variation. And when the mean over the variation, the coefficient of variation is 1, that's a Poisson distribution. And you can see that some of these ISIs fit that distribution, not perfectly, but they approach it. Whereas others, maybe looking at this, the coefficient of variation here, that's the number I'm looking at. And others are lower, suggesting that they are more periodic. So the organoid, based on the anatomical localization within the organoid, there are different ISI distributions that suggest that something interesting is going on here, even physiologically. And here's when we're looking at a whole bunch of them. Now let's look at another feature here that also is, replicates what we see in the brain. This is done by another talented, there's some great people in the lab working on this project, I must say. This is TJ Molen, who is from the Netherlands. He's a graduate student, not a postdoc. And he wanted to look at the coupling between neurons. That is, if we look at one neuron firing, what's the likelihood that any other neuron in the array is going to be firing? So we set up paired coupling between every pair of units, every pair of firings that we see on the array. Now, the time it takes for a signal to get across the synapse is 5 milliseconds or less. That's point A. We call that the latency. So if there are two neurons that are firing in relation to each other, that is, if one is activating the other, we would expect the latency to be 5 milliseconds or less. And so that's what TJ, we call him TJ, that's what he's looking at here. He sets up, like here, he's just taking unit 32 and unit 26 and using a technique called spike time tiling coefficient and looking at the strength of the relationship between these two units. Here's the math behind it. We don't have to spend too much time on that, but it's how you do the spike time coupling, spike time coefficient. And we're going to now, we randomize it to show that, you know, what we're getting, what I'm about to show you is real. And here's what you get. So once again, the X and Y axis here is the real estate. That's the space over the array. And every one of those dots is a unit that's firing. The gray lines are the pairwise connections between any two of them. And the thickness of the line is the strength of the connection. And the reason some are red and some are blue is because that particular subset, the blue, almost all of the signal coming that related to that node is incoming signal. And the red is almost all outgoing signal. And gray, which is many of them, in most of our examples, it's most of them, have a more even distribution of incoming and outgoing signals. So we have receivers, we have senders, and what we call the gray are brokers. They're the ones that are in between. And so here's what you're looking at here is the network, connectivity network in this array. And this would be very, very hard to do. It certainly cannot be done in a human brain. This would be very, very hard to do even in many animal models. It really is. Now we're getting into technology that's particularly suited for the organoid and the MEAs. So here's what I just mentioned before. The red are these senders, the two are the receivers, and then we have these brokers. And this shows the numbers, about 15% are senders, receivers 21%, most of them are brokers. Okay, so that's the directionality. And here, now I'm going to show you another one. This is another organoid, which I want to make another point. This is now, the one I showed you before was one snapshot in time. Let's see what happens over time. Here's a 10-minute recording of an organoid slice here. And what we're looking at on the right here is first off, the raster plots on the left. And now we're going to look at, here's the sum of all 10 minutes. But what TJ did was create, in B down there, create a sliding window to look at the network over time. Here's what he sees. As you slide the window along, the network is fairly stable. Look at those pictures. They look more or less the same, but they are also not identical. There is, remember, all the firing we're showing you so far is spontaneous. This is intrinsic activity in the organoid. And now I'm showing you intrinsic dynamic activity in the organoid. That is that the organoid is changing over these short time intervals some of the details of its connectivity patterns. So this suggests, I think, an extremely interesting kind of plasticity that may be operative in the organoid. Now remember, this is happening now. This is an organoid. It's just sitting in a dish. There's no body. There's no like input or output. This is all what's happening within the organoid itself. I'm going to come back to that point at the end. So now, please alert me if I'm getting if I'm pushing, we're getting toward the end. But here is, remember I told you that those gray lines, the thickness represented the strength. So let's look at the distribution of strengths within the organoid. And what you see here is this can be fitted to a truncated power law. Basically, it can be fitted in a number of different ways, but there's one big point here, which is a sort of what we call a long tail distribution. That is, there is a few connections that are very strong and a large number of connections that are quite weak. This has been described in terms of anatomical connectivity as a skeleton, now reading over on the right, a skeleton of stronger connections in a sea of weaker ones. So this kind of thing has been looked at a little bit in terms of just looking at the morphology of synapses in the brain and saying some are big and some are small. The big ones are probably stronger, and you see this kind of long tail distribution. But now we're seeing it physiologically. And I think that is really cool because that sea of weaker connections may be a source of plasticity in which the organoid, and we like to think maybe in the brain, can draw upon when it has to rewire itself with experience. So now let's talk about the LFPs, the local field potentials, the lower frequency stuff. And it's been well established in physiology for a long time that if you deconvolve the raw data, you can deconvolve it into a number of frequencies that have long been known as delta, theta, alpha, beta, gamma. Gamma gets really quite fast. It even starts to overlap with the spiking activity. A lot of delta when you're sleeping. Theta we thought was going to, oh yeah, if you take Valium, you get more alpha. The theta seems to be associated with learning. It's a little bit of a loose connection there, but there's some very interesting data that's been done in animal models, including a Nobel Prize to the Mosers, where they looked at the correlation of learning something and firing that is phase locked to the theta. So I'm going to show you what we found here. And that is now, so let's look at this picture in a little bit more detail. Here, let's go over here in the panel in the upper left. So the black is the raw signal, and then we see in the red, it's deconvolved. So we're seeing the theta rhythm, theta oscillations. And now what we're going to do is zero in. If you look over here on the right upper panel, here's the array again. All of the dots are units firing away. And we're going to zero in on neuron 1, 2, 3, and 4. And they're firing. And then now let's go down in the lower left, panel B. We're looking just in this short time area where there's gray here. Look at the scale bar here. This is 5 seconds is the black. So this is a much shorter interval down here in the gray. It's within a population burst. And now let's expand that and look over here in the lower right, where you can see the theta oscillations as you move from electrode, or I should say neuron or unit 1, 2, 3, and 4. And this is called, whoa, here I go again, coherence. That means that the wave, the oscillation is traveling from 1 to 2 to 3 to 4, that it's moving over time as a waveform. Non-stationary features of these theta oscillations. And then we go a step further. This is basically showing the same thing. So now let's superimpose the spikes onto the theta waves. And here we are, we're looking at this, let's look at this gray area again, blow it up, look at the theta waves, look at the black dots where the spikes occur. And if we do this over many, many examples of looking at this theta envelope here and look at where the firing fits, we can come to the conclusion that statistically, in these what are called Rayleigh plots, there's a statistical association in these polar graphs for the firing to occur within a certain sine angle on the waveform. And that's what all this data is. I won't go into a lot of detail here, but the bottom line here is that this really has many of the features of what's called phase locking of the spikes to the theta oscillation. In the absence of any input, that the organoid is intrinsically doing this. So we do this in another way too. There's a device called neuropixels, which Elmer Guzman looked at in the lab. Instead of putting the array down on a surface, we can stick in a CMOS kind of a pillar here that would penetrate the organoid. And right here, when we penetrate it to the depth where the firing is, you can see all these colors. That's where the firing is. Now, without slicing it, just putting in the CMOS in that manner. Well, the title of this talk was "The Organoid is a Bioprocessor." And so, in the brain, we have an input and we have an output, but in an organoid, we don't have an input or an output. But we do have this controller with these processes and possibly some sort of feedback. So here the organoid is decoupling from sensory input and motor output. And in this way, the brain creates this closed loop system, and we can look at that here in the absence of a body, which tells us what I'm about to say in the next pictures here, which are some quotes of some very famous people that have very prescient people who thought of this, before there even were organoids. So let's look at this quote here from John O'Keefe and Lynn Nadel. John O'Keefe won a Nobel Prize over here for the discovery of place cells in the brain, that is, cells that tell us where we are in space. Look what he says. A neuronal system in which activity occurs in the absence of sensation serves as a physiological scaffold capable of let me just go over this again. A neuronal system in which activity occurs in the, here's the important part, absence of sensation serves as a physiologic scaffold capable of representing information from the external environment. Another key part coming up, before it is experienced. Amazing. Then Yuri Bazaki, who currently works at NYU, wrote this in a book. The brain already starts out as a nonsensical dictionary. What a great term. It comes with evolutionarily preserved, pre-configured internal syntactical rules. That's everything we're talking about. That can generate a huge repertoire of neuronal patterns. Exactly what we're talking about here. None of these guys knew about organoids. I think what we're seeing now is the kind of activity that they were thinking must exist in the brain. But the basic concept was here even before neuroscience. This guy, Immanuel Kant, introduced this concept of synthetic a priori knowledge that is independent of experience. Of course, he wasn't thinking at all in any way that we're talking about right now, but the idea that there is synthetic a priori knowledge that is independent of experience. We're not talking about knowledge now in the organoid, we're talking about abstract waveforms, this framework. So I would say that the a priori framework of abstract waveforms can be detected in an organoid and really is what our research is right now all about. So last slide, people working on this. Most of this work was done by Tal Scharf, postdoc, he just recently got a job up in Santa Cruz, by the way, which is the goal of every postdoc. And here's the graduate student, Elmer Guzman, also did some work with us. Morgan helped a lot with the technical stuff. Stella did all the single cell work I showed you. A couple people from UCSF here, Camillini and Sri. Actually, I didn't talk about what they contributed. They looked at how you can take our data and apply it to some of the human technologies like the magnetic electroencephalography that's done on human brains and how that may relate to what we've seen. We also had help from a graduate student in computer science who's guided by Linda Petzold on the faculty of CS. Paul Hansman, physics, comes to our meetings and always has interesting things to say. And Gabe Luna did some of the staining I showed. So that's it. Thank you, everybody. Thank you all very much for your attention.
This is George Bader. I have a question. The cells that you were using, were they human?
Yes, they're all human cells. They start as skin fibroblasts, and then they're made into stem cells. And from there, they're made into neurons and organoids. So everything here we're looking at is human cells.
Have you looked at animal cells?
We, in this manner, I mean,
Yeah.
no, we have not. But other people have, and people have made organoids from chimp and gorilla and other primates, and there's, you can do this with animal cells too. We haven't done that.
Similarities or differences?
Pardon me?
Are there similarities, large differences, et cetera, or?
Well, there are.
No.
There's very little work so far at this level on the physiology on organoids from any type of cell. But when people look at the anatomy of how an organoid is made from a gorilla or a chimp, there are some differences that have begun to emerge. And they have to do with the size of the organoid, the number of neurons that are contributing to the lamina. There are some differences that are beginning to emerge.
Thank you for a wonderful talk. So back to the title of your talk about using it as a controller. Are you actually able to do some kind of computing with this? And also, what are the prospects of getting around the blood supply problem?
Yeah, well, so The title was really a future, an aspiration. We do want to use this for computing. I've been talking to a couple of people about that. I think it can be done. I think that there's a way of, what I'm very interested in doing is trying to integrate some of the features we see here, like these interspike interval distributions and some of the general features as perhaps a way of adding layers to an AI system, a machine learning or a deep neural net that we can then use for this, to use this in computing. But we haven't done that yet. It's a lot of conversations right now. To get around the blood supply is not a trivial problem. People are trying to introduce, you can make blood cells too from stem cells. You can make all the cells that are in the adventitia and the smooth vascular cells. So trying to integrate them into what the organoids that we have is an active area of research. We're not doing that, but some people are.
Follow up on that.
When you talked about the plasticity, does that mean you've seen reinforcement of connections, or does it change over time?
Well, that's what we have not, we're looking for reinforcement of connections. We're just starting that work. I don't know exactly what's going to happen. I mean, the first thing we'll look at, of course, will be reinforcement of connections, but there may be a kind of rewiring that will be of interest too. That's when we're also very interested in.
Human plasticity is, I think, I recall, it used to be thought that it was related to pruning, the process of pruning away things that are unused, and the number of cells in the brain went from about 200 billion to less than 100 between birth and maturity, or 20 years or so. Is pruning part of the plasticity? Is pruning part of the process of plasticity that you're looking at?
Well, you're absolutely right. There's an enormous amount of pruning that goes on during development, and pruning is active even after development. That is, synapses are still being gained and removed. All that's part of plasticity. So we are looking at what we're calling plasticity, but I think it would be premature for us to actually use the word pruning, because almost all of our measurements are physiologic measurements. We're not actually counting the synapses and looking at their appearance and loss. But I'd love to do that. We just are not doing that right now. Here's a question in the chat. Maybe I'll read that one. How many groups are making brain organoids, and what other types of organoids are being made? Well, this is new technology. There's not a huge number of groups doing this, but it's not a small number either. I would say around the whole world, there's probably maybe not that many groups, maybe 50 groups doing brain organoids. There may be, but for other tissues, people are doing organoids of all kinds of tissues. I just recently read a paper about somebody that's used trying to make, there's certain diseases where you lose lacrimal glands, so you can't keep your eyes moist and cry. And they've used stem cells to make lacrimal glands. You can make anything from stem cells.
I'd like to follow up, if I could, on a question about blood supply and organic computing possibilities.
Yes.
So are there still missing cell types? It was a little bit difficult to tell from the earliest slide. It looked like maybe there were some astrocyte and glial kind of cells that were missing, but in later slides, it looked like they might be there. How critical is that to getting to some kind of real useful organic computing?
Yeah, that's a really great question. We don't, well, the first part, of course, the early slides, the immunocytochemistry as opposed to the single cell, you're looking at this in two different ways. So the immunocytochemistry is not as quantitative, it's just showing you where things are. For the numbers of cells that are contributing to each of those populations, the single cell, that what we call that UMAP, that that colorful thing with all those shapes, was showing you the cells there. But I do, sort of mentally mapping that UMAP onto the immunocytochemistry staining I showed is a little bit tricky to do that, because It's just their techniques are different. But your question was really more profound. It was really how much anatomical fidelity do we need to use an organoid for computation? And I don't really know the answer to that. I think there's the fact that in even this very crude organoid, which really, still has a center that has neurons that aren't quite making it. And, there's a lot of flaws here. This is not a brain and it's tiny. But the fact that it is doing these phenomena of showing, this kind of these truncated power law distributions and networks and all of that stuff means that there might be some principles that we can look at here that might be useful for computation. And that's very much what we're looking at here. I've been talking to some physicists that want to, sort of apply kind of, some field theory. This gets out of my knowledge very quickly, but to look at some other techniques that might sort of show how you can sum up all the activity within the networks and begin to show the dynamic features. I think there's a huge amount to do here.
I'd just like to come to that one of your last slides.
Yes.
Where you were implying that the functional organization, or seemed to be implying that, or others were, that the functional organization of these organoids was approximating kind of a priori knowledge a la Kant.
Yeah, that was a little
It
bit.
just seems to me that, physiological function and knowledge are probably two very different things.
Absolutely. That's why I qualified that by saying that what we're seeing here is not knowledge, obviously. These are just abstract waveforms that when you have a body, then we convert experience into abstract waveforms. Here, that conversion is not taking place. There's obviously no knowledge here. So, the invocation of Kant may be a little loose and are provocative. I agree with you. I make no claims that there's any knowledge in an organ. And no consciousness either, by the way.
Right, good.
Should I read this question here? Let's see. When you do these experiments over and over with different organoids, what things do you see are very repeatable? What features tend to repeat closely? So this is a problem with organoids that sometimes, they're not absolutely reproducible if you're looking at the patterns of cells that I showed you in the early slides, the counting of the cells. What we're really looking for here are exactly what you're asking. That is, it's my belief, and I think we have a little evidence for this, that what's repeatable here are certain principles, that there's always going to be this kind of type of distribution of strengths, of edge strengths, and that distribution will follow maybe a more universal feature of organoids. There's going to be these ISI distributions. There's going to be phase locking. I think some of those are going to be the more universal features that are going to emerge from underlying dynamic principles. We have to take something that's changing all the time, like a brain, and figure out what is shared among all organoids or all brains.
This is George Bader with another question. How do you direct a stem cell to go in to develop into any particular organ cell?
Mm-hmm. You do it with specific. So one, I mentioned that we have 22,000 genes, and some subsets are turned on in different cell types or different organs. Organs are made up of cell types. So, and the way that happens is that there are transcription factors. Transcription factors are proteins that turn on subsets of genes. So if you introduce the transcription factors that will guide cells toward brain or kidney, that's the first step, is to take the appropriate transcription factors that are used to make a specific cell type and express them in those cells. And then the second step is to use growth factors like hormones that the cell is listening to that will then guide them even further and more precisely toward a cell type destination. So combination of transcription factors and growth factors is how we do it.
I was curious about the geometry of the slice, since you're only seeing connections between things that happen to lay in the plane of the slice. What percent of the organoid is the 500 micron slice?
It's probably about 5%, because there's something like that. But you're absolutely right. We're only seeing the connections in the slice. Clearly, things are happening in the Z, and that's why I'm very enthusiastic about that NeuroPixels, where we put the CMOS probe, which has something like, I think 520 electrodes on it, not as many, but put that in so we can now get information in the Z.
Oh. And I wasn't familiar with the UMAP. That was the unit on your graphs. Can you just briefly explain what that is?
So sure. So you have maybe 100,000 cells that have transcriptional profiles, and so you're looking at a very high dimensional space, and you're then trying to begin to do clustering by doing a nearest neighbor analysis. So now you can cluster them through that kind of analysis. But those, if you only do nearest neighbor, it's still very hard to distinguish clusters. So UMAP is basically a visualization technique that takes the nearest neighbor analysis and spreads it out in a non-linear manner to make those really nice looking clusters. You have to be careful about information in those clusters, because while they are showing us the cell types, the way they've now been shaped there is mainly for visualization. And so there's a lot of controversy about how much UMAPs will tell us. They're very good for telling us the cell types. Do they tell us anything else? I'm not sure.
You showed us some beautiful images that are taken of these materials you're working with. And I'm wondering if if you have all of the imaging tools that you would like to have in order to continue this work, or if there's certain types of measurements that you are not able to do now, but you would really love to be able to do, either electrical or visual using microscopy.
Yeah, well, wow, that question is so on target. We are salivating over the possibility of extending the electrical imaging to an optical. We're not seeing what we're looking at electrically. So we've been talking a lot about building an optical layer on top of the array, or a visualization layer in which we can now actually look at stuff. And this is really, I would say this is one of our next major steps we have to do. It's an engineering problem as much as a biologic problem. And it's important not just to be able to see the individual cells as they are, but there's a technique, which I'm sure many of you know about, called optogenetics, in which you could now shine light down on individual cells. And when they're constructed in a certain way, you can actually use light to get them to fire in a very controlled way based on the way the light is being pulsed. And so we are extremely keen to figure out how to build an optical layer. Thank you for that question. Here's a related question in the chat. Related question: what problems in your experiments are you looking most forward to solving? Well, one of them I just talked about. We really have to see what, the optical layer is very important. The other one is I'm very keen on trying to collaborate with people that have more computational sophistication than I do, to begin to how we can use this information indeed for looking at learning. Actually, I would say, to put this in most concrete terms, we would like to use the organoid to build a closed loop system in which we would actually now be able to take the information that's coming out and very, very rapidly use that information to do updates that would then allow us to direct the way the organoid is behaving electrically. Those are the two big things I'm most interested in right now.
I mean, the brain has many subsystems like the cerebellum and the cortex and so on. Is this organoid resembling any particular brain structure?
Great question. Yeah. So this brain organ, the way we do it, is mostly resembling the cerebral cortex. But people have used growth factors to direct organoids more toward the cerebellum, hippocampus. Other parts of the brain are also being made as organoids. And what you can do is you can actually take organoids that are made, that are directed toward different parts of the brain, and then put them together and they fuse. They're called assembloids. And that's another fascinating direction. I should say one more thing, because you asked about future directions. Another thing that we actually are doing is the extent to which we can actually look at neurological disease using organoids, because you can't really, you can make animal models of diseases by putting in genes that cause Alzheimer's and all that stuff, but you can't do that in humans. But we have put into organoids mutations that lead to features of Alzheimer's, and have been looking at that in the same way. And we have some papers that are currently under review and looking at how cells change when they're expressing genes related to neurological diseases.
Early on, when you were describing firing characteristics of cells, you delineated what you referred to as sending, receiving, and broker cells. And at the time, I wanted to lay that over the kind of input, output, and let's call them interneurons, for lack of a better terminology right now. But then more recently, you said that was only a very narrow slice. So maybe you're not seeing all of it, but it suggested early to me that you might be seeing the recapitulation of the layered structure in cortex and other subcortical regions as well. Is that true, or am I off the base there?
No, I think it's more or less true. We're losing. There's clearly inhibitory interneurons that are in the two-dimensional 500 micron plane. So for instance, to see local field potentials, those oscillations, you need to have inhibition. They need to get turned on and turned off. So there's clearly inhibitory function there that is operating within the plane. But I totally agree with you. I think that to get a more full picture, we're really looking at only a fraction of what's there by having a slice, if that was your question.
Yeah, and so that organization seems to go back to the previous question that I had asked about function versus knowledge. You're seeing this organization arising, let's refer to it as more or less innately, right? Self-organized.
Yeah, self-organized.
Better term, right? But maybe not too different. So the question then is, this is all happening without input. And the question obviously is, what happens to that organization when it has input? Because input occurs very early in normal embryonic development.
Absolutely.
Certainly visual, auditory, kinesthetic, possibly gustatory input. Certainly some of those organoids were approximating 7 and 8 months of development. That's well past the time when you have active sensory input. So what's the effect, and how might the organization you're seeing be kind of spurious because there's no input? Sorry,
Yeah.
long question.
No, great question. So the absence of input is even more profound than the absence of these, no eyes or ears or mouth. It's really, there's no heartbeat that's been appreciated that the fetus must be feeling. There's no, if the mother has a meal and now there's senses of hunger and, amino acids floating around. all of the lack of input is profound, and so indeed you could say that because there's no input, that what we're seeing is spurious. But the fact that it does look like some of the things that happen in the brain in terms of, things I mentioned, like phase locking, makes me think that there's something interesting going on there that really is happening intrinsically, or is being self-organized. And that's what we want to get to. I think there was another layer to your question, which I'm forgetting.
Well, that's fine. I think I was throwing spurious out as a extreme point.
Yeah.
I think it's unlikely given the self-organizing properties, but the real question is, that input clearly has a function as well as a purpose longer term, but it has a function in organization. And
Right.
right now you don't have any way to make contact with that.
Yeah. Oh, yeah. So that's where you were, right? So I think we do have a way to make contact with that, because, well, we know we can't, put a visual stimulus to an organoid. What we can do is electrically stimulate. So that's the form of input that we're going towards. So right now, what we're doing right now is to say we stimulate 3 electrodes, and then we do a time series, and we repeat those 3 electrodes, repeat them numerous times. Then recompute the network. What are the features that we're going to see as we give the only stimulus we can to these organoids? When we do it repeatedly, we do it over time, and can we guide the organoid, if we can develop a closed loop system, toward some sort of an output?
Brilliant. Go ahead.
As you think about the next steps to take in your measurements and want to use light, it occurs to me that John Bowers at the Institute of Energy Efficiency, who's studying how light can be used to increase the density of semiconductor circuits and the speed of them, is a person that would probably be worth brainstorming with about some of these issues, because I think that's a really important way to go forward.
Yeah, that's a great suggestion. I know John a little bit, but I've never talked to him about organoids yet. So would be really, really terrific. There are really, I think, if I put my list of, things that are interesting to do, I mentioned one of them, and part of that optical layer is light delivery. So, that would be really important. Here's a question. Would growing bigger brain organoids be an advantage? And if so, what limits their size? Have you considered growing them over some structure more than just a gel? Well, main thing that limits their size right now is probably the lack of a vascular supply. Growing bigger organoids, I think it would be an advantage. I don't, why? You can ask the question by having what do we get by having a big brain? It's really remarkable how few neurons you need to do a lot of the ordinary things that we do. You can, and how you can teach an animal to do all that stuff. Most of the connectivity in the brain is the brain talking to itself. Almost all the wiring is, upwards of 90% of the wiring of the brain is internal. So there's something that's going on internally that's giving us a richness that you don't necessarily see when you do these rather crude experiments by, training a primate to reach for a banana or something. So I think we could, it's going to be very hard to get at, but bigger organoids might start to get at some of these more abstract things that we do. And then the question went on to say, have you considered growing them over some structure more than just a gel? Yeah, there's a lot of interesting material science here that would be really interesting, because you can even get gels that have different, more about this than I do, different tensions that they would put on the organoid. I would love to explore materials a little bit more here. Well, I think you're on mute, or we don't hear you.
This isn't really question, but I was just struck by the diagrams of the brain and the differentiation of the cells and how they compare to an artificial intelligence, machine learning kind of neural network, and probably also being more on the scale compared to a human brain of the size of a neural network.
So in AI, the word neural network is so misleading because it really does, is so, most of the connectivity in a neural network, they talk about back propagation, things like that, but really it's mostly going forward from one layer to the next. Most of the connection in the brains is going in the opposite direction. When you talk to like first year biology students, you talk about information going from the eye back to the lateral geniculate ganglia, back to the occipital cortex. You get the impression that it's like directional, but most of the information is actually going the other way. It's going back from the occipital cortex, back to the lateral geniculate. It's these feedback systems that are really the dominant thing. None of that seems to be fully encoded yet in AI systems. But it's even more profound than that because, the biggest thing, and just another thing we're very interested in and looking for collaborators is that the brain learns things from very few examples. And we have to figure out how it does that. This is just, it doesn't happen in machine learning systems.
Great, thanks. Does anybody else have questions that you'd like to ask? Well, this has been a really interesting talk. I thank you so much.
Thank you. What a great group. A lot of great questions. Thank you so much.
Thanks. And we'll see people next month.
Okay.
Bye.
Bye-bye.